Hello, this is part 4, continuing on with macrolides. On slide 40, we looked at how they work, interfering with the process of ribosomes sticking amino acids together. On to slide 41, some examples of macrolides: erythromycin and azithromycin, and good for people who are allergic to penicillin, good against gram positives, good against this thing called atypical pneumonia or walking pneumonia. Not good against these things called Enterobacteriaceae. So Enterobacteriaceae, mentioned those before. The entero part means within or inside. These are the bacteria that live in our digestive tract in particular, and they are all gram negatives, and they have that outer membrane, and that can block the uptake or the path of macrolides into the cell. So bacteria can become resistant to drugs like macrolides, and they do it in a similar way as we saw earlier, by changing the shapes of their molecules. So earlier we had bacteria changing the shapes of their penicillin binding proteins. I guess we shouldn't say that the bacteria are choosing to change the shapes, just we have variation in shapes, and we have some shapes giving the bacteria an advantage, and those shapes are selected for. So in an environment where there's lots of antibiotics, two different shapes, one shape doesn't bind the drug, one shape does bind the drug. The shape that doesn't bind the drug is more successful, and then that's the one that's passed on to the next generation. So generation after generation, you get a shape that is better and better and better at resisting the drug. And shape is very important too in ribosomes. Ribosomes are made of proteins, and they are made of ribosomal RNA. So we have our large and our small subunits. You could imagine all the folded up proteins and ribosomal RNA. We can imagine a place, just a little box in our large subunit of a certain shape. And we can imagine our macrolides. They have a particular shape, and our macrolides can then fit into this shape in the ribosome, and then they can start to interfere with the process of transfer RNAs, binding, bringing in their amino acids, and interfering in particular with the bonding of two amino acids together. So if we were to imagine a shape change to occur in our ribosome, that box is no longer a square. Our drug is still the same shape, and now our drug cannot fit into the 50S subunit. If it can't fit, it can't stick. If it can't stick, it can't interfere with the amino acid binding process, and therefore it no longer prevents that process. No longer has any effect on the cells. On to slide 42. We're looking at other medications that inhibit protein synthesis, but they inhibit it in different ways. So we have protein synthesis occurring. We have the information in DNA being transcribed by molecules into messenger RNA. We have messenger RNA being translated by ribosomes to proteins. So far we've seen a couple of drugs that interfere at this stage. They interfere with the ribosome and they prevent the production of the protein. But there are other stages in this process, and on slide 42 we are looking at fluoroquinolones. Fluoroquinolones inhibit what's called topoisomerases, and these are enzymes that wind up and unwind DNA. So let's take a look at our DNA structure. DNA structure, as we may know, is a double helix, and on each strand of our DNA there are the A's, the T's, the G's and the C's. And in a regular configuration these strands are folded up, they're coiled up. So our RNA polymerase has to attach, and our RNA polymerase has this mechanism to unwind the DNA. As the DNA enters the RNA polymerase, it's unwound and then it's wound back up again at the end. And in this unwound space, the RNA polymerase can read the DNA and it can make its messenger RNA copy. One thing that happens in particular is that as our RNA polymerase is unwinding the DNA at this point, the DNA down here gets tightly wound up. In fact, it gets too wound. If it was to continue winding, it would break. So we have enzymes, these topoisomerases, that come along and they relieve the winding of the DNA. In fact, their job is to snip and let the tension off and then reattach so that we don't damage the DNA. So if we inhibit topoisomerases, we cause damage to the DNA as molecules like RNA polymerase unwind DNA to transcribe it into messenger RNA, and that DNA damage is then irreversible. So they are bactericidal against a wide variety of bacteria. Oops, sorry. That's how fluoroquinolones work, and one example of fluoroquinolone is ciprofloxacin. Last on our list for this section is on slide 43, rifamycins, and they also work to block, indeed they work to block RNA polymerase. So we saw our DNA passes through our RNA polymerase, gets unwound, and then winds back up at the end. And simply our rifamycins, they bind to the RNA polymerase and they simply prevent the forward progress of the RNA polymerase. So the RNA polymerase can't read the DNA. The RNA polymerase therefore can't make the messenger RNA. The messenger RNA then can't be transcribed into a protein. So by stalling the progress of our messenger RNA, we have prevented the production of a protein in this case. An example drug for rifamycins are rifapin or rifampin. That brings us on to the study guide slide on slide 44. Recall the function of ribosomes in protein synthesis. In fact, recall the entire process, beginning with a gene on DNA being transcribed into messenger RNA by an RNA polymerase, that messenger RNA then being intercepted by a ribosome, and then all of the things that happen inside a ribosome. Codons, transfer RNAs with their anticodons, transfer RNAs bringing in amino acids, the ribosome enabling the docking of transfer RNA's anticodons to messenger RNA codons, and the ribosome putting together amino acids and building proteins. And then the drugs we've looked at, aminoglycosides. These drugs stick irreversibly to the 30S subunit. They are bactericidal. They kill the bacteria. That binding to the 30S subunit is irreversible. Our tetracyclines, bacteriostatic. They can bind to the 30S once again, but then they can detach too. Our tetracycline is the one that causes teeth discoloration if given to young children. Our macrolides, those are the ones that stick to the 50S subunit, and they get in the way of the ribosome's ability to bond the amino acids together. Our fluoroquinolones, these are the ones that mitigate, they take care of that overwinding. They relieve the stress on the DNA molecule. They inhibit the enzymes that relieve the stress. Without stress relief, say after a RNA polymerase has passed through, that would cause irreparable damage to the DNA. So we inhibit topoisomerases in the bacteria, and as a result the bacteria are killed. And then our rifamycins, these are the ones that directly block the progress of RNA polymerase. They stick to the RNA polymerase itself, and the RNA polymerase can no longer progress down the DNA and can not make then a successful copy of the gene in messenger RNA.